For half a century, we've looked to neuroimaging with this huge promise that it would finally unlock the biological secrets of schizophrenia. But after literally thousands of studies, what if we actually learned for sure? In this explainer, we're going to dive deep into a major review that sifted through 50 years of research to find some definitive answers on how brain structure is different in schizophrenia.
All right, so here's our game plan. We'll kick things off with a quick look at the history of this research and, crucially, how the scientists graded the evidence. Then we'll get into the nitty-gritty, the key findings for both gray and white matter. After that, we'll explore the timeline of these changes and wrap it all up with the most important take-home messages. The story of this research is really a journey from noise to clarity. It all started back in 1976 with the very first CT reports that hinted at enlarged ventricles in the brain. What followed were decades of small, often contradictory studies. The review calls this the heterogeneity era. For a long time, people just blamed the condition's diversity for the messy results. But the real problem? Small sample sizes. Now, though, we are firmly in the big data era, with these huge collaborations like the Enigma Consortium. And this is the key. By pooling data from thousands and thousands of people, we finally have consistent findings across different methods. This confirms that these results are real, not just some statistical fluke. So, to make sense of all that data, the authors of this review had to create a really rigorous system to separate the solid findings from the weaker ones. Think of it as creating a framework for scientific truth. And here it is. This hierarchy of evidence sets an incredibly strict standard. For a finding to be labeled convincing, it has to be based on over 1,000 cases, have a p-value smaller than 10 to the minus 6, show very little inconsistency across studies, and have zero evidence of publication bias. Let me tell you, that is a really high bar to clear, and as you'll see, very few findings actually meet it. Okay, before we jump into the results, there's one key metric you need to understand: effect size, or Cohen's d. This isn't just about answering yes or no to whether there's a difference. It tells us the size of that difference. A bigger d-value means a more substantial, more meaningful difference between the groups. So keep that d-value in mind as we go through the data. With our framework all set up, let's get to the findings, starting with structural MRI, or SMRI. This is the technique that measures the brain's gray matter. Basically, it's information processing centers. So when we look at the brain as a whole, a really fascinating pattern emerges. First, there's a small but significant reduction in intracranial volume, that's skull size, with an effect size of minus 0.17. That hints at something happening during early development. But the reduction in the whole brain volume is bigger, at minus 0.30, and the reduction in total gray matter is even bigger still, at minus 0.49. So what's the takeaway? The loss of brain tissue is actually greater than the reduction in skull size, which points to something happening after the skull is fully formed. The evidence for these findings, by the way, is graded as highly suggestive. And this is where the power of that grading system really shines. After 50 years of research, only 2 regional findings meet that top-tier criteria for convincing evidence. What are they? Volume reductions in the thalamus and the amygdala, both with an effect size of minus 0.31. And look at the low heterogeneity for the thalamus, only about 21%, which means the finding is incredibly consistent across tons of studies. These are, without a doubt, the most solid gray matter findings we have. Now, moving down a tier to the highly suggestive category, we find changes in some key frontotemporal and limbic systems. The biggest regional effect is in the superior temporal gyrus, which is critical for processing sound, with a pretty large D of minus 0.58. The hippocampus, which is vital for memory, also shows a big effect at minus 0.52. And these findings are backed up by massive studies confirming widespread thinning of the cortex with a similar effect size. Okay. So this is where it gets really interesting, because we can start to connect these structural findings to the actual clinical symptoms of schizophrenia. That superior temporal gyrus, the region we just saw with the largest effect size, is heavily involved in auditory processing. And sure enough, reductions in this area have been linked to how severe a person's auditory hallucinations are. But the story of this brain symptom link is a great example of the scientific process at work. You see, early, smaller studies suggested a pretty strong connection. But then, when the much larger, more rigorous Enigma mega-analyses came along, they found that the association was actually much weaker, even though it was still statistically significant. So what's the bottom line? The link is real, but it's modest. The initial excitement was probably pumped up by publication bias, where studies with stronger results are more likely to get published. It's a really important lesson in scientific humility. All right, let's shift gears. We're moving from the gray matter processing centers to the white matter. You can think of this as the brain's structural wiring or its information highways. And to study this, scientists use diffusion MRI or dMRI. The most consistent thing we see with dMRI is a widespread reduction in something called fractional anisotropy, or FA, across the brain's white matter. Now, FA is basically a measure of how organized and efficient those nerve fiber bundles are. The overall effect size is minus 0.42, which gets a suggestive grade. And here's why that matters. This global reduction in white matter integrity is significantly correlated with cognitive problems, specifically with processing speed and general IQ. So we've established what changes in the brain, but the next huge question is, when do these changes happen? Are they there from birth, or do they get worse over the course of the illness? To figure this out, researchers look at high-risk populations. On one hand, you have people at familial high risk. These are close genetic relatives who don't have symptoms. They only show small brain differences with pretty weak evidence. But then on the other hand, you have individuals at clinical high risk who are already starting to experience early symptoms. This group shows widespread cortical thinning that looks a lot like the pattern in established schizophrenia, and the evidence here is highly suggestive. This tells us that many of these brain changes really seem to accelerate just as symptoms start to show up. So what about after the illness starts? Is there an ongoing progressive loss of brain volume? Well, this slide shows an excess annual loss of about 0.3 to 0.6% in patients compared to healthy controls. But, and this is absolutely crucial to understand, the evidence for this is graded as weak because the results are really inconsistent across studies. The current thinking is that this progression probably isn't a continuous, lifelong process for everyone. Instead, it seems to be most noticeable around the time of the transition to psychosis and might be worse in people who have poor clinical outcomes. All right, so after sifting through 50 years of data, what are the definitive conclusions? What are the key things we need to take away from all this for science and for medicine? Let's boil it down to 4 main points. First, after half a century, we can finally say with confidence there is convincing evidence for specific brain structural differences in schizophrenia, like a smaller thalamus and amygdala. Second, these findings are robust. They're not a fluke of one particular analysis method. They've been confirmed over and over. Third, the timeline is getting clearer. We know some brain changes are there before the full illness begins. They seem to speed up around that time and might continue in those with poorer outcomes. And finally, and this is a really key point for clinical practice, the effect sizes are modest. That means there's still a huge amount of overlap between patients and healthy controls, so neuroimaging is not, I repeat, not, ready to be used as a standalone diagnostic test. And that leaves us with a critical question. We know the group differences are real and scientifically validated. But because the effect sizes are modest, we can't use them to diagnose an individual. So what is the path forward? How do we take these hard-won insights from the lab and turn them into tools that can actually guide clinical practice? Well, that remains the central challenge for the next era of research.